Hello students, welcome to this series of videos about the cell. So to begin with, let us see how the cell was discovered. Now the concept of the cell was put forward first by Aristotle way back in 384 to 322 BC. However, the term cell was introduced by an English botanist, Robert Hooke, who observed cells for the first time in 1665. He developed a simple microscope under which he examined a thin slice of cork. In 1674, Antony van Leeuwenhoek, a Dutch cloth merchant and a microscopist by hobby, he designed an improved microscope. He was the first one who observed live cells, bacteria and protozoans in rainwater. He named them as animalcules. The first operational compound microscope was constructed by Zacharias Janssen and Hans Lippershey in the 1590s. Later, they designed an improved version of this microscope. So let us go through the different parts of a compound microscope. We have an ocular lens, also called eyepiece, which forms the erect image of the object and further enlarges it by 10 times or 15 times. It is inserted on the body tube. There is a revolving nose piece where different objective lenses can be fixed. An objective lens forms the initial inverted image of the object and enlarges it. Objective lenses are available in different resolutions like 10x, 40x, 100x, and many more. The stage clips hold the slide firmly on the stage. A condenser lens focuses the rays of the light on the object. It is fixed just below the stage. The light source provides sufficient adjustable light to be focused on the slide. The base gives horizontal support to the microscope on a surface. The coarse adjustment knobs are used for rapid and precise focusing on the object. The fine adjustment knobs help to get a sharp and clear image of the object. The stage provides supporting surface where glass slide is kept under the objective lens and allows light to pass from the condenser lens below it. The arm forms the vertical supporting structure of the microscope. So these are the main parts of a compound microscope. In the year 1931, a German physicist named Ernest Ruska and an electrical engineer named Max Knoll designed an electron microscope which used a beam of electrons and could magnify an object to over 2 lakh times. Hence, it became easier and possible to observe and understand the ultrastructure of cells and cell organelles. Stay tuned for all upcoming biology videos. Subscribe to our YouTube channel and hit the bell icon to get all our future notifications of videos.